Gene therapy. This topic is taken from the chapter of biotechnology and its applications. So, first of all, you should understand what is gene therapy. It is most important to know about the gene therapy. Okay, first of all, if the gene is not present in an individual or it is not very efficient, then we can replace these gene with the help of the gene therapy. This is only known as gene therapy. So, gene therapy is basically genetic or congenital diseases. Gene therapy basically deals with the genetic or the congenital diseases. So, let us consider a baby. Okay. Let us consider a baby. This baby lacks some sort of gene. So if the gene is inefficient, then we can increase its efficiency of the gene. So if the gene is completely absent, then we can insert it. This is what gene therapy. Okay. Therapy is an attempt to deal with genetic or congenital diseases. It is an attempt basically to deal with the genetical diseases or any congenital diseases. So, replacement of a faulty gene by healthy and functional gene is called as gene therapy. First of all, you should know this definition, which is very important, very easy. The faulty genes means genes which is not good, genes which is inefficient, or the gene which is absent in the person. It can be replaced by a healthy or functional gene is called as gene therapy. Very simple. Then the first disease to have gene therapy is ADA, adenosine deaminase deficiency. Here baby girl was suffering from the disease called as adenosine deaminase in 1990. Okay, was the first case of gene therapy. Very simple. Okay, leading to the deficiency of ADA and problems of immune system. So ADA deficiency can lead to the severe combined immune deficiency, which is also called as SCID, severe combined immune deficiency, which is the problem in immune system takes place. ADA deficiency can also be treated with. First of all, we should know that how to treat the ADA deficiency. We can treat ADA deficiency by bone marrow transplantation. What we can do in bone marrow transplantation? This is the first method. Here we can directly take the bone marrow and we can transplant it, and transplanted bone marrow may be having or must be having ADA gene. So it is necessary to have ADA gene for transplanted bone marrow. Then enzyme replacement therapy. So we can directly inject enzyme into the body of organisms. This is what enzyme replacement therapy. We can directly inject the enzyme into the body. Transplantation we have understood. We can directly take the bone marrow and we can transplant it, and transplanted bone marrow must and should have the ADA gene, because this is the ADA deficiency. ADA gene is must and should be required. Then enzyme replacement theory. Here we can directly inject the enzyme into the body of an organism. Then the third one is lymphocyte isolated from patient's blood are cultured in vitro. So here what happens? This is the thing, this is the cultured medium where lymphocytes are present. Okay, lymphocytes are important in creating the immune response, as we all know. So these are the lymphocytes in the cultured medium. So functional ADA cDNA are then introduced into the cultured lymphocytes. These lymphocytes are returned back to the patient's body. So these are the lymphocytes in the cultured medium. In the cells of lymphocytes, we will insert a gene. Here we will insert a gene which is basically responsible for ADA deficiency. This gene is called as ADA cDNA. We will insert it, which is functional ADA cDNA. Okay, we will insert this gene into the lymphocyte, then it will obviously start replicating, start making multiple copies of it, right? So now the lymphocytes replication starts. These are the temporary. These all processes are temporary. These three processes are very temporary because it is a continuous process. Here we have to give enzyme lot of time. here we have to give enzyme, here we have to give ADA gene. So it is the continuous process, it is not permanent. Okay, this is what happens. So these lymphocytes then added to the patient's body so that we can temporarily solve the problem of the ADA deficiency. Lymphocytes are not immortal. Lymphocyte dies, then gene also dies. ADA gene also dies because lymphocyte lifespan is fixed. So what happens? Therefore, repeated infusion of genetically engineered lymphocyte is required. As I said earlier, these processes are temporary and require the continuous process. So we have to do the genetically engineered lymphocytes. Insertion is required in such type of cases. Permanent cure. We all have understood that these are the temporary treatments. How to do the permanent cure for ADA deficiency? Introduction of a gene isolated from bone marrow cells producing ADA into cells at early embryonic stages. So we have to take the gene from the bone marrow and it will produce the ADA, which is very important. ADA gene is very important only at the embryonic stage.